Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show. Today bringing you another really awesome guest involved in creating a better tomorrow on some really interesting fronts. Today we have the honor of being joined by Dr. Eliah Overbey, who is Assistant Professor of Bioastronautics at the University of Austin, where she's involved in really pioneering research in the fields of astronaut health, specializing in spaceflight-induced genomic changes, and her work has been focusing on mapping these changes in the human body that occur during spaceflight, as well as developing these really interesting Earth-independent laboratory systems to ultimately help make humans a multi-planetary species. Dr. Overbey came to University of Austin from her previous research position as research associate at Weill Cornell Medical Center, and her most recent projects have been involved in analyzing these unique genomic changes in astronauts from missions like SpaceX Inspiration4, as well as she's been working on data analysis and sample collection for both Axiom-2 and the Polaris Dawn missions. Dr. Overbey's work also helped launch the Space Omics and Medical Atlas, a really interesting online portal containing the largest compendium of various molecular measurements from astronauts. And she also serves as a vice chair of Cornell Aerospace Medicine Biobank, which is the first major biorepository of samples from commercial astronauts. In addition to all that, she is a chief scientific officer at BioAstra, which is a nonprofit organization developing various innovative healthcare systems for astronauts. Dr. Overbey received her undergraduate bachelor's in computer science from UC San Diego, went on to do her Ph.D. in Genome Sciences, University of Washington, and then did postdoc work in computational biomedicine as a NASA space biology postdoc fellow in Dr. Chris Mason's lab. A lot to get into today, a lot of really interesting topics. Honored to have her with us. Dr. Eliah Overbey, thank you so much for taking time to come on the show.
Thank you for having me. That was a really thorough introduction.
I enjoyed reading all about you, and I'm looking forward to discussing all these topics. As we typically do, would love to start off by just handing things over to you to talk a little bit about how you got started in this, amazing journey you've been on. In reading some of the articles, I know you have extreme interest in making us a multi-planetary species. Love to hear how it all started out for you.
Yeah. So I first dipped my toes into the water of, I guess, spaceflight and genomics when I was in grad school. There was a mission that happened where an NASA astronaut, Kate Rubins, was on the ISS, and she started doing some of the first molecular biology experiments in spaceflight. Specifically, what she was doing was she ran the Oxford Nanopore MinION device. So this is a small portable DNA sequencer. They were testing it in microgravity for the first time on the space station. They prepared these DNA libraries on Earth for a few different organisms, for mice, for some microbes, and they sent it pre-prepared into space. And she loaded it onto the sequencer, and the sequencer worked perfectly fine in microgravity, which was, it's always a bit shocking when things work in microgravity just straight out of the box. But that got me wondering, how much molecular biology work has been done in spaceflight? Was this really the first time? I didn't know yet. I hadn't asked myself that question. And when I started doing research, I realized not only was this the first time that DNA sequencing had been done in space, this was really the first time or at least there wasn't any existing data sets available from astronauts after they'd come back down to Earth. And we have all of the resources available to us, from the state-of-the-art labs, these astronaut genomic measurements, mutations due to radiation, changes in gene expression, epigenomics, these just weren't being studied at all. And so that really opened the door for me, both, concerned that we weren't collecting this data, but also made me realize how much work there really is to be done in this field.
So, that leads to your 2021 dissertation, which is entitled A Characterization of Cephalid Transcriptome Changes in the Spaceflight Environment and Mapping of Epitranscriptomic Landscape of Pluripotent Stem Cells. And I found your dissertation fascinating from the perspective that, you talk about sort of the epigenetic landscape in your thesis, not just about, sort of the current sort of way we talk about epigenetics in terms of the markings and methylation and histone modifications and all that stuff. But you refer to sort of the broader, the Waddington epigenetic landscape that all sorts of things, whether it's epigenetic modification, the methylation, or whether it's radiation or microgravity or a variety of things that we're going to run into up there, all come together to these really interesting ways of stabilizing different patterns across the genome, which could do things like lead to disease, which could do things to our stem cells, which can impact ultimately when we are an interplanetary species, how embryos form in space and so forth. Talk a little bit about this work, because it's amazing work. I loved it, and I would love to just hear a little bit about the 2021, what led up to 2021, and some of the work that went into your thesis.
Absolutely. I'm glad you liked it, because it was very challenging to put that thesis together. I was doing research simultaneously in the Genome Sciences department at the University of Washington, but was also working with NASA and some of their working groups on the side and starting to look at gene expression changes in mice during spaceflight. And the astrobiology program at the University of Washington helped me through that journey. So I had my foot in two different realms. So on the one hand, I was in the Genome Sciences department, and I was doing all of this really cutting edge genomics work. So I was doing, in the thesis, I mentioned heavily epitranscriptomics. So this was using that same sequencer that's been validated on the space station, looking at whether or not we could utilize some of the unique features of that sequencer in order to detect modifications on RNA on top of that A, U, C, and G base pair set. So these would be, specifically, I was looking for M6A, which is a modification on adenosine. And so that modification, what it can do, depending on where it's at, it can increase transcriptional turnover, or it can potentially impact also alternative splicing of these transcripts. And so I was studying this in the context of stem cells. So stem cells, there's typically two different states that we look at stem cells in. There's the naive state and there's the prime state. The prime state is ready to undergo gastrulation and produce, the three different layers, the endoderm, the mesoderm, and the ectoderm during development. The naive state is not at all ready for that differentiation step. And if you try to push it down that pathway, you won't get what you want. And so the lab that I was in did a lot of work characterizing the differences between the naive and prime state, specifically at the epigenomic level, looking at differences in different histone modifications and different methylation on DNA. But they hadn't looked at methylation on RNA. So I was characterizing the differences in the epitranscriptome between these two different states. And so the reason, and it wasn't just that assay, there was also, when we look at, I was doing some Hi-C data, both the wet lab work and the lab, and also the data analysis for a method called Hi-C. And this is a method that looks at chromatin contacts. So when we think about DNA, you might think of it as being a strand of molecule that's linear, but when it's packed into your cell, it actually has three-dimensional structure. And that three-dimensional structure is going to change how the cell is regulating itself. And so I was doing a lot of work on that end as well to look at how cells, how DNA and how enhancers are regulating the genome. And then when I looked, on the other hand, to what was being done in spaceflight, I wasn't seeing any of these assays being performed, not just on human samples, but also on mice and on other organisms that had been flown on the International Space Station. And so on the other hand, I was working with NASA, who was collecting a lot of transcriptomic data from mice that had been flown on the ISS, and looking at gene expression differences. And so that's kind of how that thesis came together, was doing the really advanced thing on one hand, and then just taking what was available from the NASA end and looking at how we could, in the future, elevate that to make it more congruent with the standards in the field.
Mm-hmm. Fascinating work. Really, really an amazing piece of scholarship there. And again, for everyone listening and watching, it's available online in the University of Washington repository. And I encourage everyone interested in the sort of the space of epigenetics and transcriptomics and so forth to take a look at it. But really, really impressive. Just wondering, if we could just, take a brief tangential turn for a minute and have you sort of introduce us to the general topic of bioastronautics, because this is an area that it encompasses all sorts of things in terms of biology, behavioral, medical in general, sort of everything that's happening to astronauts, and an area that, needless to say, as we're putting more and more people up there in recent years and foreseeable future, we have to have a much more in-depth study of all these systems and how they interact in space. And obviously, it would contrast it with another topic that we have discussed a little bit on the show, namely astrobiology, which is more, I guess, involved sort of the origins of life in the universe, which may, I guess, in some ways be slightly different. But when, we think about things like extremophiles that we study in terms of radiation resistance and so forth, maybe some interesting connections there down the road when we're trying to develop these countermeasures to keep us healthy in space. Introduce us broadly to the space and sort of what the bioastronautics research community is all about in 2024.
Yeah, I like that you made that comparison to astrobiology, because when I tell a lot of people what I do, they immediately assign the term astrobiology to it. And that's not quite the right fit. Because astrobiology is, like you said, it's the study of the origin of life and also where life might exist elsewhere in the solar system and in the universe. Bioastronautics, on the other hand, is more human-centered. And it's more centered around taking humans from Earth and getting them located in other places. And this encompasses, my specific niche in this is the genomics of everything that's going on in astronauts, which is itself a big niche, but there's also psychological aspects such as confinement. There's higher order physiological aspects, there's pharmaceutical aspects, pretty much anything you could imagine that you would need to keep a human alive in space would fall under the bioastronautics category. And the way that I think about this, I break this into subfields as well, because when I think about the research that I'm doing, there seem to be two different categories things can fall into. There is, what do we need to do to optimize human health in space for it to be, for you to be as healthy and as thriving as possible? And then there's the category of, what's sort of the minimum set of things that we need to give you to keep you alive in space. And so you can approach it from two different angles depending on your mindset.
And now, connected to that, as I mentioned in your bio, you were integrally involved in the development of the Space Omics and Medical Atlas. This is located at the Mason Lab, I believe, at Cornell, and is sort of the beginnings of well, it's a major repository, but I guess one that's going to be growing substantially as we have more and more of these missions and are collecting data. I think it has the twins, the Kelly brothers twins study information, as well as things that you've collected from Inspiration4 and Polaris Dawn. Walk us through a little bit about what the SOMA database is all about and what, I took a gander at it online. Obviously, a little more too complex for me just to walk through, but take us through a little bit about what's involved in that database today and where you see that going as an important tool in all this.
Yeah, so part of the impetus behind creating that was just that there are so few astronauts flying that we're actually collecting this data from. Well, there's few astronauts flying, and of those astronauts, only a subset are participating in these omics studies. And so we really have just a very small cohort of individuals who we're even measuring this data on. And so we want to make sure that that data is accessible to people, like ready at their fingertips, so that people can access and see everything that's available, because it's really not that much at this point. The other aspect to this is that since there aren't very many people, we want to pool as many together as possible in order to get statistically significant results. So you mentioned the NASA twin study. That was N of 1, I guess N of 2 if you count the ground control Mark Kelly, but that was 1 astronaut in space for a year. The Inspiration4 mission was 4 individuals. Polaris Dawn is 4 individuals, with 1 overlapping from the Inspiration4 mission. And then Axiom-2 was another cohort of 4 individuals. So we're up to all of those cohorts together. We have around a dozen people in there. So we can start to pull together what we're seeing in the data there to make more significant results and more significant conclusions. But also I see it as a huge resource for just hypothesis generation. So if you're applying to NASA for a grant and you want to refine your hypothesis a bit more on something that's changing in the microbiome or in the immune system, you could go through and look at all the human data and see if you can find evidence to support your hypothesis when going back to NASA and asking for more grant money to run more specific studies.
Mm-hmm. Excellent. Wonderful. Anyway, so I thought, in addition to, discussing that, sort of the work at the genomics level, I thought it'd be fun to jump into a couple of the publications that you put out there. Again, these are sort of 3 quite diverse areas, but I thought it'd be fun for the audience to sort of broadly understand the scope of everything that you're looking at. Again, thinking of the bioastronautics landscape. And I thought an interesting place to start would be your October 2023 paper, Viral Activation and Ecological Restructuring, Characterize the Microbiome Axis of Spaceflight-Associated Immune Activation. And this is, this is quite interesting perspective. Obviously, we think about sending astronauts, humans, up to space, but we don't always think about the trillions of microorganisms and viruses and other things that live on and in us in various ways up there. And this paper is really interesting in that you studied, a range of things from sort of plaque formations in the oral microbiome to interesting toxin, antitoxin systems, stress responses. What were some of the interesting things that stood out to you? Obviously, the early days, but in how our human microbiome needs to be sort of deeper studied as we contemplate bringing it with us up there.
Yeah, a bit of background on that. We collected hundreds of swabs throughout the course of the Inspiration4 mission. We collected them on the ground, in flight, and post flight. And then while we were in flight, we also collected a bunch of swabs around the Dragon capsule itself. So the crew was swabbing their oral cavity, their navel cavity, nasal cavity, though they were also swabbing their belly button, behind their neck, behind their ear, their T-zone on their face, their armpits, between their toes. We got a pretty thorough landscape of the microbiome of these crew members. And so what we saw, again, as you were saying, I think it was interesting to see in the oral microbiome, there were those increases in plaque-forming bacteria, or bacteria at least associated with plaque formation. So you can see, oral hygiene is suffering a bit during spaceflight, as you might expect. There were also bacteria associated with different stress, like environmental stressors that could be placed on the skin. So you could see that the skin itself was undergoing I mean, it was undergoing the changes in microgravity, the changes in radiation, and just the normal stress of being in a confined environment. So you could see different bacteria associated with that profile. What was interesting too was that we actually collected 2 different time points in flight. So we got one near the beginning of the flight and we got one near the end of the flight. And we actually analyzed, what was going on. Was there anything different about each of these crew members? And we actually, during an event where we had all of them together, we gave them what we called microbiome awards based on unique features about each of their microbiome during the flight. So Haley Arsenault, for example, she was solid as a rock. She had the most consistent microbiome. Jared was the most Spartan. He had the fewest number of microbes on him at any given time. Cyan Proctor, Dr. Proctor, she was Ellis Island because we saw the most influx of microbes onto her skin between time point 1 and time point 2. And Chris Zimbrowski was the most Martian because we found the most unknown fragments of DNA on his skin that we couldn't map to any existing organism in our database.
Very cool. You're mentioning the skin. When I read Scott Kelly's book several years ago, I remember, at the very beginning, I think, maybe the day after he comes back from the year in space, he's talking about how he literally wants to scratch his skin off because of all the changes, as you were just mentioning, not just microbiome, but all sorts of other things. And you published also in 2023 about this in February, More Than a Feeling: Dermatological Changes Impacted by Spaceflight. Again, here, not just the microbiome, but DNA damage and repair dynamics, mitochondrial dysregulation. What are some of the interesting things you're finding out in terms of the skin? And obviously, we'll talk about countermeasures and sort of dual use later on, but clearly, a few of us are going to experience that environment, but obviously, some of the things you learn and the countermeasures you will develop for space maybe have really important applications here in terms of skin health.
Yeah, so that skin health was a really interesting factor. We wanted to look at that more in the Inspiration4 crew. And so we actually collected, this paper is not out yet, we collected skin biopsies from the crew. So we took a small piece of their deltoid before and after flight, and we performed spatially resolved transcriptomics on that. So that was using the NanoString genomics platform. And what we could do with that was we could actually, or at least how that platform works, is you'll take, a layer of this tissue that you've collected, you'll mount it on a slide, you'll do some staining with it so you can identify the different regions of that tissue. And then you put this probe set over it. This is the NanoString platform. There's this probe set that'll bind to the RNA. And then based on where you shine the UV light on that slide, the RNA will be released from the slide. So you can actually prepare separate libraries from very specific regions of the skin. So using that method, we were able to get different libraries from the outer epidermis of the skin, the inner epidermis, the dermal tissue, and then regions near vasculature, blood vessels in the skin. And looking at those different transcriptional profiles, what we saw was we could actually see more inflammatory pathways being triggered in the epidermal layers than in the dermis and the vasculature regions. And we could also see more DNA response, damage response in the outer layers compared to the inner.
And one more I'd love to touch on, and this is a kind of recent paper in April of this year, genome and clonal hematopoiesis stability, contrasting with immune circulating free DNA, mitochondrial and telomere length changes during short duration space flight. Obviously, these are all let's say components of something that we profiled a bit on the show, namely the hallmarks of human aging, and this is obviously another kind of crucial dual use theme. You're going to be learning a lot of things up there that impact the health span, whatever you want to call it down here. What are some of the interesting things you're learning at this point? Clearly, in this paper, not everything lasts long. Some things persist, some things do not, like the telomere elongation. What are some of the exciting things you're finding out in some of this research?
Yeah, so one of the things you mentioned in there was clonal hematopoiesis. And so one thing that we look at in these astronaut crews after they come back down is we look to see in their bloodstream if there's any particular mutations that are taking over the population of cells that could be indicative of cancer. So fortunately for this crew, we haven't seen any evidence of clonal hematopoiesis occurring. So those 3 days in flight, those were relatively safe. And then you mentioned the telomeres as well. So the telomeres we collected from Inspiration4. This was something that was noticed on the twin study when Scott Kelly was up there for a year. It was observed that telomeres in certain subpopulations of PBMCs were getting longer during spaceflight. And I think there were certain articles online that called space the fountain of youth because your telomeres were getting longer, but that wasn't, the correct interpretation. But we were very curious in seeing if this holds true, because Scott was up there for a year. The Inspiration4 crew was up there for 3 days. They were up higher than the International Space Station, so they were getting exposed to more radiation than you would on the ISS for 3 days. But they still were not exposed to anywhere near the amount of radiation as you would for a year on the ISS. So there was this question in my mind. Like I know on Earth, high intensity interval training has been associated with telomere elongation. And so I was wondering, since astronauts are working out for 2 hours a day every day on the International Space Station, if that might have something to do with why their telomeres were getting longer. The results from the Inspiration4 crew make me believe that that is not the case. Because what we saw with the Inspiration4 crew was every single crew member's telomere length was trending upwards just during these 3 days in flight. And for one of those 4 crew members, it did reach the threshold of statistical significance. So that indicates to me that there's something about microgravity and the space environment that's contributing directly to telomere length, because they weren't exercising in that cabin. If you've seen the inside of a Dragon capsule, it's about the size of a large camping tent. You're not really doing any exercise routines in there.
Little bit about the work that you're doing and can continue to do in terms of Earth independent laboratory capabilities, because clearly there's a wealth of stuff here on the terrestrial side, but as we get as you and you talk about this on your website, from low Earth orbit to the moon to Mars to wherever else, we got to have really cool systems that are smaller and compact and can allow us to do the work, whether it's gene sequencing or cellular culturing or whatever the thing may be out there. Talk a little bit about some of the things you're going to be working on in terms of Earth independent laboratory capabilities.
Yeah, so there's one project I've been working on for a little over a year now, trying to get DNA sequencing to work in a minimal resource environment. So if you want to do a DNA sequencing project in space and you didn't have very many resources you could bring with you, how would you be able to execute this? So I'm looking at things like if there's no centrifugation, if there's no cold stowage, if there's no heat block, so I can go over a certain temperature. So like what I'm doing to heat certain sample steps, or the sample up during certain steps, is to actually use like friction from your hands and see if there's a protocol that we can do where that generates enough heat to facilitate some of the adapter ligation steps for library preparation. And so this is a protocol that, we have a version 1 of it, me and the few people in the lab who've been developing this, that we're currently, looking for a mission to fly it on. We have some leads, so we might be able to make some progress on that very soon. Still just tackling the DNA sequencing and flight steps. Because I did all this, like I mentioned Hi-C and more epigenomic work that I was doing in grad school, and then you switch to trying to do this stuff in space, and it's back to square one. You have to go back to the fundamentals, and the most basic thing that you can do. It's this question of how can we just get, strands of DNA to sequence through a nanopore. That's the validated device for in-flight. And there's probably, I think there would be a lot. It's small and lightweight. The other systems are very large and might have some fluidics issues. So the Oxford Nanopore MinION system is really the system to do spaceflight sequencing with at the moment. And so we're really just trying to figure out if you want to collect a sample from a human and get it on the sequencer, how are you going to sequence those strands of DNA? It's back to square one.
What's going on at BioAstra?
Yeah. So BioAstra, we formed a little bit over a year ago. I was looking for a venue to help make the spaceflight studies that we're doing more reproducible across the community. So going back to that issue of we just don't have that many people going into spaceflight participating in these studies. If you're a researcher and you do get an opportunity to collect blood or urine or these other biospecimens from these astronaut crews, how are you going to figure out, or if you want to compare them to previous studies, how accessible is it for you to figure out how everyone's done these in the past? Because you could do your experiment, you could collect it from your crew, you could just compare it to those crew members, but that limits the impact of the analysis that you can do. Whereas if you can compare it now to every single crew that has previously flown because you're drawing in the same type of blood tubes or you're preserving urine in the exact same way, suddenly the statistical analysis that you can do on your data becomes a lot more powerful. And so we do, as part of publishing our scientific results, we have method sections where we say exactly what we do. But sometimes if you've ever read through a method section, it's not complete, steps are left out. It's not always the most accessible depending on, if it's open access or if it's behind a paywall. So what BioAstra is doing and why I was interested in it is that we're actually taking everything that's been done on these experiments and we're bringing them into the light and giving people step-by-step protocols that they can follow to make it much easier to reproduce existing protocols. So I was really interested in it from that angle because I want to be able to do the best, have the biggest sample size that we can for these analyses. My CEO, Savi Glow, she comes at it from another angle where she looks at the healthcare system in the US and around the, anywhere really. I don't know if anyone's really solved the issue of healthcare, but she sees spaceflight as a way of rebuilding that system from the ground up and imagining what it could look like if you do get the opportunity to start building that system from scratch instead of having to make amendments to the current system that we already have. And so, she's coming at it from this big picture approach, and I'm coming at it from these building blocks from the bottom up. And we're meeting in the middle to put together this system where if you're an astronaut or if you're managing astronauts going into space, how are you going to see your health data? How are you going to access that data? And how are you going to see the research level information that you may be participating in as part of your flight? So that's that's in a nutshell the broad strokes of what we're trying to do. We've also brought on recently another twin. So we had a twin approach Chris Mason about doing another twin study. His name's Commander Brent West. He's a professor at the Naval Academy, and he's really interested. He has a twin. He's really interested in going into space. And so now we're also looking at how we can commission a twin study 2.0 mission. And so what we're doing for this is we're looking at this from scratch where, if you're a group of civilians and you want to go to space, say like the Inspiration4 mission did that, what is that pathway that's available to you? So we're working on forging a repeatable, reproducible pathway for groups of civilians to access spaceflight and participate in scientific research.
Again, it's early days here, but are you thinking a lot at this point about sort of as I don't know what the proper term is, but sort of the bioastronautical countermeasures that whether that is repurposing interesting drugs here on Earth for benefit in areas like radiation exposure, studying extremophiles and understanding how they do what they do in nasty environments. What are some of the things that get you excited in terms of after you have developed this database, this wealth of information on human health and space. In terms of what happens next in the coming years, whether that's for the moon, Mars, and beyond, what are some of the dream areas that you want to, start looking at in terms of, again, making us multiplanetary or healthy multiplanetary species?
Yeah. So thinking forward to those countermeasures, I know there are companies looking at those drug targets. I'm thinking a step ahead and trying to
Okay.
think of how are we going to execute that clinical trial. So once you have that drug target, once you have that drug you want to try, you don't just want to give it to one astronaut and see if it works in them. You want to actually be able to execute a more systematic approach. So that's part of going back to BioAstra. That's part of why we're trying to find pathways to do these missions independently of NASA, so that there is a way for once we have a drug target, once we want to be able to say, that this drug works in astronauts. If you're going to Mars and you want to protect yourself from radiation, you should take this drug. To really be able to push that, you're going to want to run a clinical trial first and be able to scientifically prove that. So I'm trying to figure out how we can execute those types of studies and the steps we need to get us there.
Mm. Clearly the big announcement that you're going to be leading this amazing set of research now at University of Austin. That was just out in the press a couple of days ago. What else is happening in terms, clearly these are the early days, but what else do you have planned for the rest of 2024? Other interesting initiatives that you could tell us about, or conferences that are coming up that you're going to be attending? Anything else in the bioastronautics community that's happening that we should know about? Please.
Yeah. I'm glad you brought up the new position at UATX that I have here. I'm really excited about that because the structure for the student curriculum at UATX is really interesting in that the last 2 years of the student curriculum here is actually dedicated to projects, with the hope being that students can actually get an entrepreneurial pursuit off the ground and turn that into a business, turn that into a company. And the reason I find that particularly suitable for the work that I do in bioastronautics is that when we go into space, when we go to the moon, when we go to Mars, I don't want it to be another camping trip. The Apollo missions were absolutely incredible and groundbreaking, but the issue was we never really repeated them. There wasn't any financial incentive to, I think, is a big reason for why that never happened. And so being able to actually make spaceflight economically sustainable is going to be really important for future exploration. And so being in an environment where you can actually get students to think about spaceflight in ways that create businesses and create economically sustainable participation, I think that's really going to push the needle forward in terms of permanent space habitation and permanent exploration initiatives. In terms of some future things on the horizon for the rest of the year, BioAstra is actually starting a podcast called Star Life that should be released sometime. I think our first episode is coming out sometime within the next week. Depending when this airs, it may already be out. You can check for it on Spotify. It's called Star Life, and it's hosted by the CEO, Savi Glow. She interviews all sorts of people across the space industry. She interviews scientists like James Green, who led a lot of the Mars rover exploration initiatives over at NASA before he retired. She's talked to Tom Marshburn, who was a NASA astronaut and is now the chief medical officer at Sierra Space. So I think if you want to get some more insight on some of the key players in the space community, particularly as it relates to putting humans in space, that'll be another great resource.
Awesome. I'll put that in my playlist, because that's I clearly enjoy this topic, but I only I'm decent at talking about sort of this segment of it, so that's going to be very interesting to learn about everything else that's happening in the domain. So no, that's happy to hear about that as well. But no, really, really exciting times. I'm excited for you. I'm excited for your team, and just wishing you the best with all of it as you get this program up and running and growing it, and hopefully we can do a follow up next year to see where everything's going. But I'm again really really awesome set of research that you've been up to, and I just love to see it translating in this fashion and moving it forward in this era where we are becoming a multi-planetary species. So I'm very excited. Um,
Yeah, a fun
Again,
fact is we've had a sustained continuous human presence in space since the International Space Station launched. That's been constantly manned. So we've had humans consistently in space for over 20 years now.
yeah, yeah. For for everybody that is going to be listening to this particular episode of our show across the various podcast networks, or who will be watching on our YouTube channel, again, you've been spending time with Dr. Eliah Overbey, Assistant Professor of Bioastronautics, University of Austin, also Chief Scientific Officer at BioAstra. Eliah, I really want to thank you for taking the time out of your schedule to come talk to us for a little while, to update us on everything you've been doing. Obviously, thank you for what you do in making us a multiplanetary species. And ultimately, as we like to say on our show, thanks for helping to create a better tomorrow for all of us via what you're up to there. Really a great story, and I look forward to staying in touch and following what you do.
Great. Happy to be here. Thank you for the invitation.
Great having you.